Hello students, in this lecture I am going to discuss about the rest of the recording of bioelectric potentials. In the previous lecture I discussed about the electrocardiogram and electroencephalogram. In this lecture I am going to discuss about electromyogram as well as the other prominent recordings of the bioelectric potentials. So let's start with electromyogram. So basically what is electromyogram? It is the recording of the bioelectric potentials, or you can say the summation of bioelectric potentials. It is the recording of summation of bioelectric potentials obtained from the muscle fibers. So what happens, each muscle fiber generates action potential, and when number of muscle fibers which are generating action potentials, they reach the surface of the body, the summation of that action potential, when we record it, we get the electromyogram. So this is in this slide you can see how the electromyogram is getting recorded using the surface electrodes. So electromyogram can be recorded using the surface electrodes near the muscle of interest. Suppose you want to measure the bioelectric potential or the muscle activity of a certain muscle of a muscle of interest, then you will place the surface electrode over the surface of the body, over that muscle, so that you can get the recording of the bioelectric potentials of that particular muscle. So either electromyogram can be measured using the surface electrodes by putting the surface electrodes on the body, or it can be measured by using the needle electrodes. That is, needle electrodes can be made to penetrate the skin and into that muscle of interest whose recording we need, whose bioelectric potential recording we need. So in this way we can measure the electromyogram. Now what happens, as the surface electrodes, they when put over the surface of the body, they can pick up the potentials from all the muscles within the range of them. So within the range of these surface electrodes, whatever potentials, bioelectric potentials are there from all the muscles, they can pick up that. Now what is the problem? What is the problem in that? The problem is the potentials, when this surface electrode is used for measuring the bioelectric potential from smaller muscles. Now what happens at that time, the potentials from nearby larger muscles may also interfere with the attempt to measure the EMG from smaller muscles. So what is happening, if I'm trying to measure the EMG, that is electromyogram, from smaller muscles, I'll place a surface electrode on the surface of the body over that muscle. Now the property of surface electrode is that it will pick up the bioelectric potentials near to its range. So it is possible that it can pick up the bioelectric potential from the nearby larger muscles, which we do not need actually. We need the bioelectric potential from the smaller muscles. So this is the problem with the surface electrode. Now to overcome this problem, what is the solution? We can use needle electrodes that can be inserted directly into the muscle of interest. Now the amplitude of measured EMG waveform is the instantaneous sum of all the action potentials generated at any given time. And because these action potentials occur in both positive and negative polarities at a given pair of electrodes, so they sometimes add and sometimes cancel, and thus, as you can see in the next slide, and therefore the EMG waveform appears very much like a random noise waveform, with the energy of the signal is a function of the amount of muscle activity and electrode placement. So this is a typical electromyogram waveform. It is just like the random noise waveform. So this was about electromyogram. Now proceeding towards electroretinogram. Basically, what is electroretinogram? It is the recording of the bioelectric potentials, or you can say the summation of bioelectric potentials obtained from the retina of the eye. And this is recorded, we get this bioelectric potential or the change in bioelectric potential from the retina of the eye as a response to the visual stimulus. So this is how electroretinogram is getting measured. So it is the recording of bioelectric potentials obtained from the retina of the eye. Next that I am going to discuss is electrooculogram. So what is electrooculogram? It is the recording of bioelectric potentials that is generated by the movement of the eyeball. So this electrooculogram potentials, they are generally picked up by small surface electrodes placed on the skin near the eye. One pair of electrode is placed above and below the eye. As you can see, one pair of electrodes is placed above and below the eye, and this is placed why this is placed above and below the eye? So that they can pick up voltages corresponding to vertical movements of the eyeball. Another pair of electrodes is positioned to the left, as you can see here, the left and right of the eye, to measure horizontal movement of the eyeball. And the recording pen is generally centered on the recording paper. Here we have the recording paper, and the recording pen is generally centered on the recording paper corresponding to the voltage changes accompanying it. So this was about electrooculogram. So what is electrooculogram? It is the recording of bioelectric potentials which is generated by the movement of eyeball. And the last one that I am going to discuss is electrogastrogram. So electrogastrogram, it is basically this pattern, this waveform, or this recording. It is associated with the peristaltic movement of the gastrointestinal tract. Now you might be wondering, what is peristaltic movement? Peristaltic movement is what? When you actually swallow food, it is the contraction and relaxation of the food in the esophagus and the food pipe, and the food is forced down the tract to the stomach. So the pattern that is associated with this movement, that is relaxation and contraction of the esophagus or the food pipe, this movement. So the potential that is related to this movement, when it gets recorded, we get electrogastrogram. And what is gastrointestinal tract? I have mentioned that it is the peristaltic movement of the gastrointestinal tract. So what is gastrointestinal tract? That is the organs that the food and liquids travel through when they are swallowed, digested, absorbed and leave the body as feces. So all the organs that they fall under the gastrointestinal tract. And for example, it includes mouth, pharynx, esophagus, stomach, small intestine, large intestine, etc. And this is how electrogastrogram is getting measured, using the surface electrodes over the surface of the body over this gastrointestinal tract. So this was all about the recording of bioelectric potentials. So I am ending this lecture here. Thank you.